Thank you very much for the opportunity to give this seminar. Actually, I had recently given a variant of this seminar at a conference in London about 3 months ago, which was specifically directed at stuff happening in developing countries. And GBS is probably not new to many people in this room. But what I'm going to try to do today is to sort of share with you some of the ways in which I think GBS has significant potential in developing countries, particularly in livestock improvement. So I'm going to sort of get started by giving you a quick overview of the love affair that we have had with livestock for coming on about 10,000 years now, and how efforts to improve livestock have undergone a very long history until the birth of the genomics revolution roughly about 25 years ago. And that technology has rapidly moved and has brought us almost to the edge of where we're basically rewriting the rules of how genetic improvement is being done. So livestock agriculture goes back principally to around the dawn of the late Iron Age / the New Stone Age. So about 10 to 12,000 years ago was when humans began to domesticate livestock. So this came a little about the same time that crops were domesticated. And as humans moved from place to place, it was easier to take their livestock with them, but also carry their seeds as well. But livestock were mobile, but plants are not. And so the intimate relationship that developed between humans and livestock is somewhat of a unique nature compared to, well, aside from the fact that they were also animate, you know. And so if you go through the literature, what you will see is some ranges for when, you know, different livestock species were domesticated. But there's a general consensus that most of this domestication was centered around the Levant, primarily present-day Iraq, Turkey, parts of Palestine, all across to the Indian subcontinent in the case of cattle. Actually, in the case of cattle, there are 2 sites which we think are independent origins of domestication. There's still some unresolved issues, but enough evidence exists to conclude at this point that there was one site somewhere in Iraq, present-day Iraq, and there was another site somewhere in present-day eastern India. And so if you go back even to the time of, you know, ancient Egypt, the role of cattle and livestock in general had become well established, as you can see in these hieroglyphics paintings. So we've been dealing with these guys for a long time. So animal agriculture provides some, I would argue, some unique advantages and attributes that, you know, have made human existence very doable. They provide a diversity to diet that until very recently you would classify out almost as a luxury. Because animal agriculture provides products and goods that traditionally have always been considered non-average. In fact, we actually see that happening in many developing countries today. One thing that has happened in the last 25 years as the middle class has exploded around the developing world is this incredible increase in the desire to consume animal products. And so I'll give you two examples. The largest pig breeding company in the US was recently acquired by a company in China, Smithfield Premium Genetics, based in North Carolina. It's not a Chinese company. And the reason is actually quite simple. The Chinese consume a lot of pork. And so if you're going to increase the production of pork, what you want to do is to acquire the company that owns the genetics. Because the genetics of many of the modern domesticated livestock is proprietary. The companies that do the improvement of these animals, just like in the case of plants, the information is not in the public domain. This is proprietary company information. So they're not just going to hand it over to you. So what you do is you buy it. And this is where I think you will see how important GBS potentially could be as we go along. I always like to share this slide when I give a talk about what I do because it's so easy not to realize what is really at stake. In fact, I read an article last week that because of the Great Recession in 2008, some of the gains we made in the last decade, we've actually lost ground a little bit. Some 250 million or so have actually fallen back again into poverty and are trying to climb their way out. So by the time I finish my talk, somewhere around 700 kids would have died of hunger and starvation around the world. So this has some real consequences. And I always remind myself about why and what we're doing, so that it's not just that it's meaningful from a scientific point of view, but this is something that has immediate and real consequences for the lives of a lot of people. If you look at how the world population is growing, it shouldn't surprise you that the largest challenges of food security and hunger, malnutrition, and so on and so forth is also highest in developing countries. And if you look at the world population projected out to 2050, you will see the purple part of this graph is primarily driven by what is happening in developing countries. I'm originally from Nigeria. The population of Nigeria grows at 2.9% per annum. That's a lot. That's a lot. In my lifetime, I'm 45. In my lifetime, the population of my country has almost doubled in my own lifetime. Because in 1970, Nigeria was estimated to have something like 85 to 90 million people. Today, it has 160 million. So this is a major problem. And if you overlay this picture and the picture I'm going to show you next, what you will notice is that the countries that have the biggest problems of food security, well, also are the countries that have the largest population growth. And so it's like being on a treadmill. You can't stop. You actually have to continue running. And so we need ways. We have to find ways to do things, smarter, to do things cheaper, to do things in a way that can get us to our destination with the least amount of time and effort. So focusing on livestock production itself, you can see that there's been an enormous increase, in the last few decades. And all of this increase is driven by developing countries. Because in the developed world, many things are mature. They're no longer increasing. In fact, every major company in the agro-allied industry, specifically in livestock, which I'm most familiar with, have all their growth coming out from abroad. companies like Genus PLC, that ABS Global, so these are companies that do a lot of the genetic improvement in domestic livestock. Their markets in the US are saturated. In fact, the only way they can make money in the US is to steal market share from their competitors. But if they go out to Brazil or to China or to Nigeria, they don't have to be stealing anything from anybody because it's all wide open. And so let's talk about cattle for a little bit, because this talk is about cattle production. So these guys showed up in the fossil record about 60 million years ago. So they've been around a while. They were domesticated, like I said, roughly around 10,000 years ago, give or take, because of their unique ability to convert stuff that we can't really use into stuff that is eminently nutritious and desirable. And in one form or another, cattle impact the nutrition and livelihood of almost everybody on the planet, except for the small population that are strict vegans. Unless they don't also wear shoes or use belts or use certain pharmaceuticals. It's very hard to not have this species impact your life in one form or another. So they're very important. I put up this slide so I can give you a little bit of the biology behind this, because I'm going to talk about this again when I start discussing the results of the GBS analysis we did. So domestic cattle are divided into two broad groups, Bos taurus and Bos indicus. And they look like this because this collection of tissue that forms the hump at the back of these guys at the bottom of this slide is what visually differentiates the Indicine from the taurine groups. Although primarily these guys are found in developing countries, there are taurine, species in the tropics. For instance, Ndama is a cattle breed that is found in sub-Saharan Africa. It's actually a taurine breed. And there's a lot of controversy as to how and why. In fact, Ndama and Muturu are two breeds found in sub-Saharan Africa that I'm aware of. There may be others. But in generally speaking, Bos taurus are typically found in the temperate parts of the world, the northern hemisphere. And Bos indicus are generally found in the tropics and subtropics. And I think that partly comes from the fact that they are very heat tolerant in general. So it would make sense that you find them in hotter parts of the world. So the official scientific name of domestic cattle is actually Bos primigenius. The taurus and indicus are kind of additional appellations. And because primigenius is common to both of them, in general usage, we just drop the primigenius and call them Bos taurus, Bos indicus. So in 2009, it became one of the domestic animal species that was sequenced. It was actually sequenced and released in 2004, but the paper officially didn't get published until 2009. So the bovine genome is roughly the same size as the human genome, roughly 3 billion letters. Anybody who knows anything about genomics knows that these kind of numbers are constantly under flux. So 22,000 was what was in the paper, but I think it's probably fewer than that now, because I don't know of any case where the number of genes increases. It's always been that the number of genes, decreases. Because I still remember when I was in school, in fact, when I was taking genetics classes in graduate school, the textbook said the human genome probably has 100,000 genes. I still remember that very well. And about 5 years later, the genome sequence was actually published. It was barely 20,000. So I think this number is probably fewer than that. So about 14,000 orthologs, they showed to be conserved across 7 mammalian species. 1,200 of them or so are, at least for now, are not found in marsupials or monotremes. So these will be animals like kangaroos and platypus. They are not placenta mammals, but they are mammals. So there are two kinds of mammals. There are placenta mammals like us and cattle. There are non-placenta mammals like platypus and kangaroos. So they either give birth to their young, very, very undeveloped, and then nurture them like in a pouch, like kangaroos do, or they actually even lay eggs. But they're still considered mammals. Genes that are involved in metabolism are some of the most conserved genes between cattle and humans. All right, so I'm going to now focus on improving livestock because at the end of the day, that's what my talk is about. See, the traditional platform and paradigm for how livestock improvement is done in the modern day is like this. So at the very top of this pyramid is the elite herds that actually drives genetic improvement through the entire system. So this will be people, or rather, farmers or entities that raise purebred animals, and they continually select these animals very intensively. They don't sell to consumers because you don't consume their animals. They sell to those who now produce what you and I consume. And so generally, most of this usually happens in selected farms. So they are not usually everywhere. And the multiplier farms is kind of where a lot of the realization of that genetics is expanded before you end up with commercial producers who actually raise what you and I buy at the store. So for example, in the case of cattle, it would be like feedlot operators, for instance, who can just go around and just buy cattle from everywhere and ship them to somewhere in Kansas or Oklahoma and raise them for 6 to 9 months and then take them to meat packing plants. And this system has worked pretty well because it depends on collecting a humongous amount of records. This is what primarily drives this thing. It's all record-based, which is why doing animal breeding is pretty much like doing statistics. In fact, at this university, you couldn't get a PhD in animal breeding when they used to have animal breeding unless you minored in statistics. Because you dealt with so much data that you almost had to be a statistics minor. In fact, up to this day, a lot of records still get dumped at Cornell from dairy farms across the country. And so these records include things like pedigree records, performance records, records on contemporaries, and so on and so forth. And then we use all kinds of statistical things to sort of crunch all these numbers together to sort of figure out who are the best animals from this whole bunch, who can now be selected to be the parents of the next generation. That's how classical animal breeding is done, as well as plant breeding. And we've done pretty well in the last few decades as this system matured. You can see the kind of progress we've made happen in roughly 40 to 50 years in most domestic livestock species. So it means that this system works very well. I mean, for instance, the amount of meat per ton of feed fed to pigs have basically doubled. It's gone up 100%. We've increased milk production per cow per lactation from about 6,000 to 10,000 kilograms. So these are very impressive improvements. But the problem with these improvements, vis-a-vis the crisis we face, is we don't have this amount of time. Because by 2050, 2.5 billion more mouths needs to be fed. And some experts are actually predicting that we need to increase food production by 70% because we need to make up for the shortfall we have now, then account for the increase of 2.5 million more mouths. So we cannot do things the way we've always done them and hope to meet this challenge. So this is how our progress this is how it has happened. So you can see how much time has been involved, a long time. So herd books were kind of formed in the 1800s. Gregor Mendel came up with his laws of inheritance in the 1860s. Genetics was named in 1906. And then population genetics, the golden age of population genetics in the 20s and 30s, when all the things about allele frequencies and linkage and all that stuff were all worked out. And finally, in the 1960s to the 70s was the golden age of quantitative genetics and livestock improvement. See, this was where things like restricted maximum likelihood, best linear and bias prediction, some of the modern tools for doing genetic evaluations were all developed, some of which were actually developed on this campus. And in the early 2000s, genomics exploded, and genomes were being sequenced left, right, and center. And now we have reached a point where we actually need to start using that information for improvement. But there was a problem. The problem was when you sequence a genome, it's just letters, okay? It doesn't really give you much. In fact, it's not even hypothesis-driven in most cases. You just get a genome, you do the lab thing, and then you just churn out letters, and they're sitting in a computer. All that information has to be used in some way, has to be converted into something that's useful. Single nucleotide polymorphisms is almost become synonymous with genomic selection, as you will see. And it's just a single base difference between any two individuals. And because they are so abundant in the genome, they have become an extremely powerful tool. And so technology ramped up on how to identify them and use them. This was something that was not even accounted for when the Human Genome Project was launched in 1990. When the Human Genome Project was launched in 1990, the plan was to sequence the human genome in 25 years at a cost of roughly $3 billion. As it turns out, the technology of sequencing improved so quickly, much faster than people had ever anticipated, that we actually sequenced the genome sooner than expected and under budget. But even more than that, once we realized how powerful SNPs could be, the technology of SNP identification and genotyping went up. In fact, when I was in graduate school about 15 or 16 years ago, I still remember that if you want to genotype one marker then, actually it was sequencing. The unit cost was really high. It was very high. It was as much as $2 per genotype. Now a SNP genotype is almost gone to zero per unit cost. Almost, it's close to zero. Because I was at a meeting in the UK a few months ago where I didn't know this before I was at that meeting, that they can now sequence the human genome for roughly $600. Roughly. So if you do the math, it means that the cost of one base is almost nothing. And so we have finally we have been ushered into an era where genotyping cost is almost vanishingly small, as it were, per unit cost of production. But the phenotypic information to utilize those genotypes have become the issue, and I'm going to say a little bit more about that. So in 2001, Theo Melwison and his colleagues published this paper in Genetics. This is the paper that got it all started. This is the paper that sort of laid the groundwork for genomic selection. It was actually a simulation paper. And the summary of this paper, which is I just recopied, is actually right here in the abstract, concluded that selection on genetic values predicted from markers could substantially increase the rate of genetic gain in animals and plants, especially if combined with reproductive techniques to shorten the generation interval. Quite simply, if you could genotype animals for lots of markers or plants, you can collapse all that information into some kind of, summary of their genetic value, and you can use that to predict their future performance, even if you didn't have any phenotype on them. As long as you had some phenotype from somewhere for which you have established a prior relationship. That's simply what it is. And the reason this is extremely powerful in developing countries, I'm going to elaborate more on this shortly, but the reason it's very powerful is because if you remember the pyramid I showed you before, I said you need a lot of records to make that system work. You need pedigree records, you need phenotypic records, contemporary records, all kinds of records. But in the developing country setting, sometimes that's a big challenge. There are no records, or the record is scanty, or maybe not even accurate, or you have records on very few animals, and yet you want to improve the population. And so genomic selection can help to identify the best animals based on genetic merit, as long as you're utilizing total genome information. Of course, it can reduce generation interval because you can capture total genetic information from day one, right? So the animal is born today, you take DNA and you genotype, and you can have the prediction applied today, long before the animal is old enough to give you milk, or long enough before the animal even reaches puberty. And so you can be making decisions today, which previously you had to wait many, many, many years. In fact, it's even much worse in a species like cattle. Because, for example, your average bull doesn't become an elite bull until almost a decade after it was born, under the old system. Because you had, especially those used for dairy production, right? So they are born, they reach puberty, you use them to inseminate females, the females are pregnant for almost a year, the females produce a calf before they can give you milk. You collect the milk from those females, and then their daughter, I mean, come on. You're talking 6, 7, 8 years into the future before things really fully mature. And all this while, you are feeding these animals, you're taking care of them, you're spending a lot of money all this time. And genomic selection is basically rewriting all of those processes. And so typically, this is how the thing works. So you phenotype and you genotype. Actually, let me show you the next slide. It's a lot easier here. So you have a reference population where you collect both genotype and phenotypes, okay? And then you can use that information to create prediction equations. Basically meaning you know what sort of relationship exists between your genotypes and phenotypes. And then you can select candidates for which you have known genotypes, but you don't have phenotypes. And then you can use those based on the breeding value that comes from that genotypic information. Although every few years, you will have to sort of reestablish that prediction equation. Every few years, you have to do that because the genome is not static. Things kind of shift around, and relationship between QTL and things break apart, and all kinds of things happen. So every few years, you have to do that. But at least you can be making decisions way ahead of time. So a few years ago, we became involved in GBS. And a group here at Cornell had published the paper below. And so we decided to apply this to cattle. And this is an extremely powerful technique because it's all out there, it's all open source, which was what really drew us to it, and which is what is drawing a lot of people to it. And this year, they finally published the paper that sort of lay out how you do this, right? And some of the people who did all this are actually in this room. So if I say anything, I think they're going to take me up on it. If I say anything that's not true. Anyway, so this technique is a technique that is not proprietary, as far as I can tell. And if you use SNP chip, it's proprietary. If you want to use AFI chip, for instance, you have to buy it from them. And it's expensive. In the case of cattle, the 50K chip currently costs between, well, it depends. A number of factors depend on whether you go through the NBCC, which is the National Beef Cattle Evaluation Consortium, and how many samples you're doing, and a number of other factors. It can go anywhere from $50 to $150 to genotype one animal, which is expensive if you're going to do hundreds or even thousands, as sometimes is the case. So this is our paper that actually applied GBS to cattle. So I'm just going to quickly walk you through what we found. So our objective were to basically take this technique and see how it will play out in cattle, and then compare that to the 50K. Because at the time, the 50K was your, kind of your, what's the word? Was the gold standard, as it were. I mean, the 770 had come out, but that was so super expensive. Very, very few people were even using that, unless you had a massive grant from somewhere. Because it was like almost $250 to do one animal using the 770K. So that's what the chip looked like if you get it from AFI. I'm not going to spend too much time on this, but you can see all of that in the paper. But basically, it was a technique that allows you to interrogate the genome by reducing the complexity of it using restriction enzyme to do partial digestions. And once you do that, you are able to, instead of doing this, you can sort of find and identify large numbers of markers across the genome, which is the goal for genomic selection. Now, the goal here, what you're trying to do is, genomic selection is your destination, but to get there, you need large numbers of markers. There are two ways to do that. You can buy a chip off the shelf, or you can find the SNP yourself and do the genotyping, which is what GBS does. It finds the SNP and it genotypes everything in one single pass, in one single action. And a lot have been said about all the advantages of GBS, so I'm not going to spend too much time on this. It does require the use of adapters and that allows you to do the sequencing. After you digest your DNA, it's broken up into little pieces, and each of those pieces have to have adapters so that you can distinguish them from each other. And then it involves multiple cleanup, steps and determining fragment sizes and all of that. You make libraries, all the standard stuff you do if you're going to do anything on an Illumina platform. So when all of this is done, you can thank the people in Buckler's group who have developed this pipeline. And you can go there, and if you have enough bioinformatics skills, which they are very good at working with you, or if you have a nice, decent grant, you can actually pay them. There are people who can do this for you. So what we found from our analysis, we used 47 animals, representing 7 different breeds. So about half of those breeds were breeds from sub-Saharan Africa, and the others were from here. 66 million reads in total at an average of 1.4 million reads per animal. The total number of SNPs was a little over 60,000, but the usable number after we went through all the multiple filter steps and call rates and so on and so forth came down to about 51,000, which is really close to the 50K, which actually contains 54,000 SNPs. And what it took to make those chips, because I was pretty familiar with that whole process, it actually began in 2005 and finally was released in 2007, Not only did it take so long, it cost a ton of money to make those chips. And by the time the chip was made and characterized and became available, it was very expensive. A lot of people use them, but the people who have used them the most are actually companies that can afford to genotype, or breed associations that can afford to genotype tens of thousands of animals. This was the number of reads and call rates for every individual animal in that experiment. And below is after we sort of adjusted for things. I think you will agree that, it's pretty decent, considering the fact of where we're coming from. So the average core rate, so the things that are really important here are things I put in red. The average core rate was roughly 86%, and I think that's pretty decent. We had half of those with a core rate above 90%. In general, in these kinds of things, you like to see 90% and above as much as possible. I mean, things that are below 90% are still useful, but you like to see above 90%. SNPs tend to be somewhere around gene-rich regions. And I think that may bring up some interesting ideas that we are following up. Average heterozygosity was highest in Brangus because Brangus is actually a composite breed from Brahman and Angus. I think 5/8 and 3/8. One interesting thing that happened in this experiment was the ability of GBS to actually identify or detect copy number variation. So there was this gene on chromosome 18 that was reported to contain a polymorphic copy number variant. And in our analysis, we actually found evidence that GBS could detect that CNV as well. Now, scalability for something that you're using that you hope to save money on becomes a major issue. And as you can see on this slide, if you run the library, see, you make libraries once, but you can run them multiple times. So a lot of the cost is front-loaded because making libraries are relatively expensive. But once you make the libraries, they are sitting there in a freezer somewhere, and you can run them multiple times. And we actually compared, this data actually came from Sorghum Bicolor, which I think they haven't published this. Sharon told me they hadn't published at the time. But by comparing that with the bovine data we had, if you run the libraries multiple times, you can actually get more SNPs without having to do, essentially just by just doing more sequencing. And as I told you before, the cost of sequencing is becoming so cheap, and I think it's going to get even cheaper. So that sequencing things will become the cheapest part of doing genomic selection. In fact, we're almost there now. So in general, like in every GBS experiment, SNPs can be identified and genotyped. But more importantly, there's very little ascertainment bias, and you don't need a reference genome. If you have a reference genome, there are some advantages, but if you're looking at the premise of what I'm discussing now in a developing country setting, where you either don't have a reference genome for your favorite species, or where you want to save money, you don't want to use proprietary technology, then GBS can fill the gap. And African breeds, this is just a side, show the lowest heterozygosities because many of them have not undergone, they don't have the same population histories as some of the breeds in the West that have undergone a lot of things through selection and crossbreeding and so on and so forth over the last few decades, or even the last couple of 100 years since those breeds were formed. People have started applying GBS to understanding genetic diversity. A lot of that is going on now. Before now, microsatellites were the sort of marker of choice if you wanted to do diversity studies and understand population structure and so on and so forth. But GBS is sort of democratizing the ability to use large panel of SNP markers. Because until now, it was really expensive to use proprietary techniques to do that. So what we conclude is that looking at cost, which is our primary focus in this study, you can do this much cheaper than you can use a proprietary 50K chip, for instance. We actually compare the cost with the 50K chip. There are low-density panels out there now. In fact, there's a 3K low-density panel used in cattle now. So if you run a lot of samples on one lane on the Illumina machine, you could potentially have the equivalent of a low-density panel. And you can reduce the cost even further. Because right now, I think the absolute cheapest you can probably do 50K for is around $50 per animal, give or take. And imputation, which is another whole thing that we didn't go into in this talk, you can use, when you get SNP prime markers from GBS, this can be used for imputation quite accurately, and I think a couple of studies have actually demonstrated this. So you can imagine a situation where you genotype parents at high coverage, and then genotype their offspring at very, very low coverage, and use imputation to fill in the gaps. And you can do this by totally avoiding proprietary technology. And you can even do this if you have no reference genome. And you can do this with little or no ascertainment bias. For those who may not be aware or understand what I mean by ascertainment bias, when these SNP panels are made, the SNPs are discovered in specific breeds. In the case of 50K, I think there was virtually no breed from sub-Saharan Africa included in those panels. In the 70K chip that came out a few years later, they tried to expand the coverage. But in the 50K, it was primarily 2 or 3 or 4 breeds, definitely less than 5. And so if you go use that chip in a breed where the breed did not contribute to the SNP discovery, you can see some issues with not finding everything that you're looking for. So I think with all the advantages that we find for GBS, primarily the cost component, I personally believe that this has a lot of promise for doing genomic selection in developing countries. Because genomic selection allows you to start with a population-based sample for which you may know nothing about their history. You may even have no pedigree records whatsoever. Well, those are all the good news. How about all the bad news? Well, cheap is still a relative term. So there's still need for resources to do this, even in developing countries. When I said it was going to be easier, I didn't mean that it will cost nothing. There's still problem of infrastructure to collect phenotypic data. You may not have pedigree records, but you need phenotypic records. I mean, after all, you are trying to improve a trait, and you need information on that trait. And so we have a unique situation now that people are calling the phenotype data collection problem, which means genotypic data is now easy. If you hair samples, some mucus, can get DNA from anything now. Within a couple of weeks, you can have all the DNA sequence you want. But how about collecting milk records? How about collecting health records, disease-resistant records, growth records, all those other stuff? That's now becoming the issue. Capacity to implement this is still a problem. Because as you can tell, this is still a highly technical thing to do. And people need to be trained to actually do this. Some of the things that I think many people have been talking to, and I talk to a lot of people because I travel a lot, is to find ways to combine efforts of both the public and the private sector to try to implement GS in developing countries. Because in developing countries, many of them don't have the infrastructure that might be taken for granted in this part of the world. And it's not really clear that, or what it might even take to build those infrastructure from scratch. And so many people are grappling with multiple approaches of how to go about this. And I think ultimately it's going to require a combination of efforts from different players and stakeholders to be able to do this. And speaking of multiple players and stakeholders, we have started talking with some donors about actually applying GBS in West Africa. And so there's this project that we're currently trying to put together. Part of it is already funded by the Bill and Melinda Gates Foundation. Is to apply GBS to indigenous poultry populations. Because these animals have largely been unimproved. Virtually no records exist on them. I mean, these farmers, they don't keep any records on them. They're just like all over the place in their backyard, scratching the ground and generally taking care of themselves. And by the way, I was born here, somewhere here. That's actually my state. My state is called Edo State. So we hope that we will get this project funded so we can try to accelerate the pace of improving these local chickens. And then we can go from this to this. Thank you very much.